as Charles indicated, we were just on the phone with the company Macrogenics about a combination targeted inhibitor immunotherapy trial for neoadjuvant GBM and at the stage of recurrence. And I want to give a little bit of background and rationale for that study. So this is a study that was done in collaboration with Tim Clousey and Rob Prinz, but also Jennifer Wargo, where the goal of the study really was to do a sort of microanalysis at multi-omic analysis of the GBM microenvironment in comparison to melanoma, to compare and contrast a immunologically hot tumor and one that is challenging to get hot under very similar therapy conditions. And so the melanoma stuff was in collaboration with Jennifer Wargo, and this is work done by Yue Liu and Alphonsus Ng in my group, and Joe Beecham from NanoString is very helpful in this analysis. Okay, so I don't think anyone needs to, here in this audience, needs to be refreshed on this, but this is just the unique character of GBM, and this is from work that Ilya Shmulevich from my colleague here at ISB published several years ago on the immune landscape of cancer, where most cancers tend to occupy multiple of these immune type phenotypes, but GBM is just sitting here in this lymphocyte depleted phenotype here. And so the goal of the earlier goals in trying to do immunotherapy in GBM, especially with checkpoints, was just to try to see if you could convert a cold tumor into a hot tumor. And work that Tim and Rob Prinz and their colleagues did, and this just compares the two studies that we'll talk about. They looked at on the order of 20 patients with recurrent GBM, and looked at there was a transcript signature that they could associate with turning a tumor from immunologically cold to at least lukewarm. And they found what they describe as a molecular non-responder and a molecular responder. So two differential transcriptome signatures, and they did see some survival benefit, but I think the real goal was to try to see if you couldn't heat up the tumor. And of course, in melanoma, that's an easier thing to do. And this is a very similar study that Jennifer Wargo directed. And here's basically the analogous one for the GBM, neoadjuvant anti-PD-1 and metastatic melanoma. So the way that we analyzed, we looked at just a whole bunch of tumors from both of these types of studies at different stages of treatment, and with both transcripts and proteins at a spatial resolved method level. And we used this NanoString a digital spatial profiling system that I think people are pretty familiar with. It's what it allows you to do, is to identify regions in the tumor based upon, say, multicolor fluorescence staining done side by side with an H&E type staining. And then to, within this sort of region of maybe this size or maybe just a little bit smaller, so this is not single cell, analyze for a panel of reasonable number of proteins. These are largely immuno-oncology proteins, or one can also capture the whole transcriptome. This is not, from the transcriptome point of view, it's not a whole genome sequencing approach. It's basically, you're looking at specific transcripts, but you can build almost as many probes as you want, but you can't see mutations. And just to show you sort of how this works and the sort of fidelity of the data, here we've taken in a series of, I think, probably one or two tumors, but we've looked at regions that are immunologically active. At least there's a lot of CD3 plus cells here, and quiet, and then looked at these basically immune signatures, CD11, CD4, CD8, etc. And just you see alternating between immunologically active and quiet, you can really tell the difference. And so this allows you to then think about the molecular signatures you're measuring these tumors within a spatial context, which I think is, the whole point of this type of microscopy. And so when you look at the whole transcriptome, and this is just a lot of data, but I think over here I want to direct you. You do see that, in fact, the molecular responders and non-responders in this sort of UMAP representation really do distinguish pretty well in a much larger analysis than what one captures with the with just a limited a transcriptome assay, such as what was used by Rob and Tim, but nevertheless gives you a consistent story. And this is unpublished, but we did do now the protein work, and that is published. That came out last year. And so here's one of these one of these melanoma tumors. And here you see a extremely robust immune compartment, very, very different from what you see in GBM. And then one can begin identifying markers that illustrate the differences between these compartments, and then distinguish those markers based upon patient response or non-response. And that's the kind of thing we did. And I'm not going to go through in great detail, other than to say that the markers that one sees for melanoma make complete sense. And when you look at melanoma, it turns out, for example, beta-2M, which is associated with antigen presentation, can really fully separate responders from non-responders in this type of analysis when you're looking in the immune-rich regions. So when you look in the GBM tumors, it's very different. In fact, there is not much that really distinguishes between responders and non-responders, except for that one does see elevated Ki-67, so cell proliferation. And I want to emphasize that because you actually will see elevated Ki-67 in both the immune-rich and the immune-poor regions, but it is statistically elevated for the molecular responders in the immune-rich regions, and is statistically elevated for the molecular non-responders in the immune-poor regions. And so the point is that it provides context for interpreting this type of cell proliferation that one sees, whether it's immune cell proliferation, good, or cancer cell proliferation, not good. And so we took that type of data and we tried to use it to predict what do we see in terms of CD8 T cell infusion into these tumors. And when you look for in the melanoma case and you look in the GBM case, one sees a level of consistency that makes you think that even though these are very different tumors, this type of analysis is giving you at least a reasonable, I don't want to exactly say apples to apples, but a reasonable comparison across tumor classes. Okay, so back on this Ki-67, the Ki-67 associated with cancer cell proliferation predicts the non-molecular responders in GBM. It's the cleanest predictor of who's not going to respond. And when we then say, well, what is it that it most associates with the Ki-67? What we see is, and I think I'll just pop to here, is we see this marker here, B7H3. And B7H3 is at the transcriptome level, it's actually pretty broadly expressed in many cells in the body, but it doesn't get translated into protein almost anywhere except for in tumor cells. It's pretty highly elevated in about 60% of cancers. And so it seems like a pretty good cancer target here. And IDO1, which I think one might have expected to pop up as well, also pops up. And so there's been a little bit of recent work on showing that this is maybe not such a bad target as well. There's chimeric antigen receptor T cells directed against B7H3 in both patients here, and this is against in neurospheres and model systems. And so this is the suggestion that we have. And if there's feedback out there for the suggestion, I'd love to hear it. I think this is a trial that we're in the process of planning, and hopefully we'll be able to execute this within, a year or so. And that's my short talk. But I'll indicate that we're working now with Macrogenics. We just had this call with them to begin taking their antibody against B7-H3 and coupling it with anti-PD-1. And with that, I will conclude, and thank you for your attention. Happy to answer any questions. I think I took
great talk, and you bought us 10 more minutes, so thank you.
Yeah, I was hoping to get feedback, but if the conference is too short or
Can
too
I think it's a dumb question. Ki-67 increased in those regions is associated with proliferation of tumor cells, right? Or are you talking about proliferation of lymph T cells? It's tumor cells, right?
Yeah, so when you have a molecular responder, you see Ki-67 associated with the immune cell rich regions. When you have a molecular non-responder, you see Ki-67 in the immune cell poor regions, tumor cell rich. So the context in which you see it is everything.
Got it.